a minute. Okay, thank you everyone for joining us to this webinar session. So today we have another great talk by Salma from Virtual University of Pakistan, and she will talk about pathway network analysis of genes involved in dominant syndromes. So thank you, Salma, for being here with us and for being part of this great initiative of webinars organized by ISCB Student Council. And the floor is yours, Salma, and please go ahead.
Okay, thank you, sir. First of all, I thanks to the organizer of the webinar of ISCB to give me opportunity to share my piece of work. Today, I'm going to talk about the project which I have completed at my master level under the supervision of Mirza Jawadul Hasnain. The title of the project is Pathway Network Analysis of Genes Involved in Dominant Syndromes. Let's start. Introductions. Genes work in group to follow some particular pathway. In order to express itself, the alteration of these genes lead to various genetic disorder. But in this study, we focus on the dominant syndrome to find out the core genes and their relationships with these multiple syndromes. Hearing is a complex process. Various cellular and molecular mechanisms are involved in it. There are various multiple dominant syndrome which give rise to syndromic and non-syndromic hearing loss, deformities of facial features, craniosynostosis, dysfunction of the ear, deformities of the bones, and various skeletal anomalies. A research problem consists of multiple dominant syndrome, round about 60 syndrome, in which a large number of mutations of the genes are involved. The autosomal dominant hearing loss is one of the phenotypical effects which is observed in these 60 syndromes. The methodology used in this research project consists of three bioinformatics tools. To find out the functional protein association network, we use string database. It is a search tool of retrieval of the interacting genes. Uh, it is a web source. It is a web source of known and predicting protein-protein interaction to find out cellular enrichment and component biological enrichment process. We use David database, which is used to find out the biological and cellular function of the genes. For gene network, we use IPS software, which is used to construct the gene network pathways and functional disease on the basis of RNA sequencing. We explored 119 genes by gene mining. When we upload our 119 gene in the string database filter out 115 genes. String database shows a very significant interactions among these genes, but there one query was arised that all of these interactions are not experimentally proven. There are only 57 genes which shows very significant interaction and experimentally proof one interactions. And these 57 genes are gene of interest in this research project. In order to estimate the signal transmission in the cell, we use David database. When we upload or filter out 115 genes in David database, here we found it present in particular three pathways which have different gene ontologies. The David database also analyze the biological pathways which describe the which genes and subgroup of the genes are present here. Here we found that our 57 genes are present in particular biological pathways having different FDR values. We use IPA for construction of the gene network. IPA is a web-based bioinformatics application and it allows researchers to analyze the data from high throughput experiments, high throughput experiment on the basis of the RNA sequencing. When we upload our 57 genes in the IPA extract only one gene which is acting gamma 1, and rest of the 56 genes are analyzed by IPA. IPA construct three networks. All of these networks are involved in the auditory diseases. These three networks consist of 35 molecules, but their number of focus genes are different. Here we can see that all of these three networks are involved in the auditory disease on its top one. They are also involved in the neurological disease, hereditary disease, and so on. We select the network one, and rest of the two network are rejected. Here we can see that our network one shows a very significant interactions between molecules and our focus genes. Here we can see that the central node, extracellular regulators kinase are connected with 20 edges, and PLC gamma, PLC gamma and are and 7 nodes. On IPA disease and functions, we see that our focus 30 among our focus 13 genes, there are only 11 genes which are associated with auditory and vestibular system development and auditory disease. In the development of the ear and the morphogenesis of the ear, gap junction beta 6 genes, fibro growth factor receptor 2, split like transcriptor factor 1 and 6 homeobox box 1 genes are associated. On the molecular activity predictions, we see that our focus genes and the molecule, our focusing and molecule shows more confident inhibition and activation predictions. Activation, but here are we see only six homeobox 1 gene and spelled like transcriptor factor 1 shows no inhibition and activations. All of these genes are involved in the auditory diseases and autosomal dominant hearing impairment. Also, they are involved in the ear development and embryonic development of the vestibular organs. So here we found that the genes which are involved in the development of the epithelial tissues are fibroblast growth factor receptor 3, 2, 1, fibroblast factor 1, and melanocyte inducing transcription factors, and rest of the molecules are also cooperate these genes. So we found that in the genes of the fibrous growth factors are also involved in the development of the external and inner ear. We also found that these genes are involved in the development of the ear and morphogenesis of the ear at the embryonic level. Uh, Here IPA analyzed that autosomal dominant deafness and digenic non-syndromic hearing loss are due to alteration in the gap junctions beta protein 3, 6, 2 and homeobox 6 homeobox 1 gene. Connexins protein are also involved in the digenic non-syndromic hearing loss. Our focus genes, our target genes are also involved in the hearing loss and development of the sensory organ, which these genes are FGFR, fibrobus growth factor 2, 1, 3, and gap junction 6. We also found that these genes are involved in the hereditary hearing loss and dominant non-syndromic hearing impairment. The gap junctions beta proteins, melanocyte inducing transcription factors, and fibroblast growth factor 3 also involved in the sensorineural hearing loss. As our focus is to determine the genes which are associated with hearing impairment, so our network one shows more significant interactions of those genes which are involved in the hearing impairment. On upstreaming analysis, basically upstreaming regulators means upstreaming transcriptional analysis that explain the target gene in the dataset. So here we found that our 11 target genes are present in the upstreaming analysis. Most of our genes are present in the plasma membrane, but some of them are nucleus on cytoplasm and extracellular space. Finally, it is concluded that there are only 11 central genes which are growth factor receptor 1, fibro growth factor receptor 2, fibro growth factor receptor 3, fibro growth factor 10, gap junction beta protein 2, gap junction beta protein 3, gap junction beta protein 6, melanocyte inducing transcription factor, and protein tyrosine phosphate non-receptor 11, 6 homeobax 1, and splat-like transcriptor 1. All of these have significant role in the hearing impairment which are associated with multiple dominant syndrome. Um, Also, in the biomedical arena, genetic diseases are treated by genome editing, such as Cas9 system. Also, stem cell therapy consider a great hope in the treatment of the hearing impairment. I end up my presentation by the saying of the Ronald Reagan that stem cell research can revolutionize biomedicine more than anything since antibiotics. At the end, I especially thanks to our supervisor who guide us at every step. And I once again thanks to the ISCB forum who give me opportunity. Really, it's a great event. Thank you.
Okay, thank you, Salma, for your great talk. It was really interesting talk. So let's start with the questions session. We have one question from attendees. So the question is, Salma. Pradeep say, thanks for the great talk. Salma, is IPA a commercial software?
Yes, sir.
Okay. You want to talk more about the IPA software, or that's?
Basically, IPA is a web-based applications.
Okay. Okay, cool. Okay, thank you. So, Salma, I have a question for you.
Yes.
It is possible to distinguish in your networks what kind of interactions are experimental based or non-experimental based? Do you have experimental validation from those interactions? Because many of these
Yes.
databases have different channels or different evidences to infer these interactions?
Basically, strain database.
For example. Yeah, but I mean, are you going to filter these networks by the evidence? I mean, if it's co-expression, if it's experimental based.
Yes, sir.
I
Your
mean,
co-expression stuff.
in terms to rank, because you are proposing next steps in your work, and maybe an interesting step is filtering the networks based on the evidence.
In this research study, we only find out the genes that are central genes which have significant interaction to the hearing impairment. On the next level, we can perform the in silico analysis of the respective genes.
In silico, you mean structural
Yes,
approaches?
sir. Yes, sir.
Oh, okay. Thank you, Salma. Nice. Okay, I think that we don't have any questions from attendees. So once again, thank you, Salma, for your great talk, and please take care. Thank you.
Thank you,
Bye-bye.
sir. Bye bye.